Welcome to the second session of Quick Biology. Kindly like, comment, and subscribe to motivate me to do more of information sharing. Today's session is going to be all about X chromosome and Y chromosome. We all know they are called allosomes. Y chromosome is smaller in size, having about 60 million bases and coding about 60 functional genes. X chromosome is 3 times longer than the Y chromosome, having about 165 million bases and coding 1000 functional genes. In today's session, first we'll see the regions of Y chromosome, then we'll see what is dosage compensation, which is a striking feature of the X chromosome, and finally we'll see some examples of genes that are present in the X and Y chromosomes. Y chromosome is divided as the pars region or the pseudo-autosomal region located at both the ends of the chromosome. This pars region occupies about 5% of the total Y chromosome. The pars regions of the Y chromosome only can synapse or recombine with the pars regions of the X chromosome during cell division. The pars regions of the X and Y chromosomes are homologous, meaning they have the same genes in them and hence can recombine to produce variation in the offspring. Though the lengths of the X and Y chromosomes are very different, the chromosomes condense or unwind to facilitate this crossing over between the pars regions of X and Y chromosome. Hence, this region is called pseudo-autosomal region, meaning they recombine similar to the autosomal or body chromosomes, which extensively recombine during cell division. The next region of the Y chromosome is called non-recombining region or the NRY region. This occupies the remaining 95% of the Y chromosomal length. Since this region of the Y chromosome does not recombine, the genes in this region is exclusively transferred from father to son and then to grandson. These genes that are passed by males onto their sons are called Hollandric genes. As this region does not recombine, it acts as a marker tool to study human evolution. The gene for hypertrichosis, that is excessive hair on the external ear, was thought to be a Hollandric gene. But today's studies are disproving it. NRY or non-recombining region is further divided as heterochromatin and euchromatin. Heterochromatin is inactive region that doesn't code containing repetitive sequence, and euchromatin is the active region that codes for functional genes confined to maleness. Within the euchromatin of the NRY region, there is a region called SRY region or sex determining region of Y chromosome, which is responsible for producing a protein called testis determining factor that initiates the formation of male sex organs. The SRY region genes start secreting TDF factor even when the fetus is 6 to 8 weeks old in the mother's womb. The SRY gene screen was used for gender verification in Olympics until 1992. Nowadays, hormone level testing qualifies one to compete in female events. That's all you need to know about the Y chromosome. A brief summary about the Y chromosome. The chromosome is acrocentric, meaning the short arm of the chromosome is really short and not clearly visible. Then the first region is pars region, which is seen at the ends, both the ends. You see the pars region or the pseudo-autosomal region, which is capable of recombining with the homologous X pars regions. Then you have short arm euchromatin, which has SRY or sex determining genes that code for TDF or testis determining factor. Then you have the centromere. Then again in the long arm you have euchromatin, which codes for male reproductive structures and hormones. And in the long arm there is a non-coding heterochromatin region. Catch you with questions on Y chromosome and more in the next session.